So my talk is actually quite a bit related to the last talk of the morning session, except that I'm going to talk more about sort of understanding the mechanism behind memory consolidation processes. Okay, that's my phone, I think, making that noise. Yeah. In fact, even some slides I have would be a little bit overlapping with what was shown in the previous talk, but I'm going to look at this from somewhat different perspective. So as we already, it was discussed this morning, the sleep is quite complex phenomenon, actually, and it's involving lots of important functions, and one of them potentially is memory and learning, consolidation of memories. And if you just look at the sort of structure of the sleep, it's interestingly very characteristic and very stereotyped across different species, from humans to some low vertebrates. And what is shown here is sort of basic sequence of sleep stages, which is repeated in humans and high animals, usually 5-6 times during night. When the brain goes through stages, it's right now those stages kind of decided to be combined. So it's typically called stage 1, stage 2, and stage 3. And stage 3 is deep sleep. So stage 2 is stage 3 called together not a rapid eye movement sleep, non-REM sleep. And there is a period of REM sleep which is quite different from non-REM, and what is shown here is this sequence of those stages happening during night. Is going from stage 1 to stage 3-4, back to REM, again down to stage 3-4, and that's repeated again and again. One interesting and really not very well understood feature is that early in the night we mainly have slow wave sleep. And lots of discussions today in the sense of role of sleep and memory and learning was about slow wave sleep. There is a REM sleep which potentially also play an important role in consolidation, and it's quite different from a slow wave sleep. It's actually much more like a wake state from basic electrical activity of neurons. And how it's also clearly involved in lots of functions, but it's much less well understood what's going on during REM sleep in respect at least memory and learning. Now, in my talk, I'm going to discuss some sort of characteristic electrical activities found during stage 2 and stage 3-4. And yeah, this is stage 2, 3, 4. And so here is one of them. It's called sleep spindles. It's probably one of the, well, not the oldest, but this is one of the first studies which describes them back close to almost like 80, 90 years ago. So this is what called spindle, some rhythmic activity which is coming in frequency 7 to 14 hertz. It's coming in events. They come and go, and they found again through this stage 2 sleep. And as we, yeah, this is another slide. This is kind of intracellular recordings which shows this event in its sort of full beauty. It's recordings from one cortical cell and 2 thalamic cell. And why thalamic cell? Because we know that those rhythms are generated in thalamus to a large extent through interaction of some intrinsic currents and cell types in thalamus. So this is a very characteristic sort of spindle. Now, as we go deeper to non-REM sleep, we get in more and more events which looks like what is shown here, called like sequence of up states and down states. And the first appears like isolated down states, but as sleep progresses to stage 3-4, it's become almost periodic in a sense that there is up state which lasts about 100 seconds, it lasts about 500 to 1 milliseconds to 1 second. And there is a down state which lasts about a few hundreds milliseconds. And again, this, what is interesting thing about those events, the whole brain does it. So in a sense, you think about this for a couple hundred milliseconds, nobody really firing in your brain when you're in deep sleep, which is somewhat scary, because something needs to start it again. So what is known that this rhythm is produced by cortex, and the mechanisms of this rhythm have been also studied, and we did some contribution. And again, it's very global phenomena, and some would say that it's very basic state of the brain in the sense that, for example, during sleep deprivation, in a subject exposed to sleep deprivation, you start to observe those events almost in a wake state. And if you do something with cortex, like isolate piece of cortex, so it becomes silent over time, it again develops and starts to doing something like what is shown here. So somehow it's very important for brain. Now, so again, I talk about role of sleep and in memory and learning, and specifically role of sleep in memory consolidation. So just to summarize, there are different types of memory. There are declarative memory, which are essentially memory of facts and events. And there are more more specific, more fine divisions, but I'm not going there. And there is a procedural memory, also called non-declarative or hippocampus independent memory, which is memory related to generally in skills like playing piano, riding bicycle, and so on. This is another version of the same sort of diagram. Now, there are a number of studies which suggest that specific sleep stages may play somewhat specific role in consolidation of different memories. This is from Sarah Mednick paper, which shows that for declarative memory, slow wave sleep is particularly important. Declarative memory, the typical experiment was shown here was subject was normal, healthy human subject, learned some, let's say, word association or some other task like this, and then naturally, kind of memory of that would be declining, and then the subject experienced some nap, period of nap, and having slow wave sleep actually led to less forgetting, so it's improved memory. On the other hand, REM sleep in this particular study didn't show much contribution, but it seems to be playing important role in consolidation or improving non-declarative memory. So if you're learning playing tennis, potentially it seems like having REM after that period would be important. Now, that picture is somewhat simplified because now there are lots of studies which shows that, for example, REM sleep can play an important role for declarative for procedural memory, sorry, for non-declarative for procedural memory, like is shown here. So it's not black and white, but essentially there are some features of different sleep stages which seems to be favoring particular types of memories. This is just one study of many which shows that somehow trying to manipulate sleep rhythms can affect memory. So in this particular study, also from Mednick lab, they use zolpidem, which basically are known to increase spindles through effect on GABA-A signaling. And what was shown that in subjects which have been using that particular pharmacological sort of intervention, memory was generally better after a period of sleep than in control. So in general, very oversimplified possibly picture of how memory sort of consolidated in a human or animal brain can be particularly declarative memories can be summarized here. So there is a period of time when there is a sort of sensory flow going through thalamus to the cortex, and cortex actually interacting with hippocampal network, creating some sort of temporal representation of the memories. So this structure potentially very plastic. It can rewire easily, and it stores that image of immediate sort of memory events. And then during sleep, when there is no information flow, this cortex and hippocampus sort of interact, and in a sequence of events called replay, and during that time, those memory traces are mapped from hippocampus to the cortex to form stable representation. And one idea why it's important to do this, because exactly Friesen mentioned earlier, we want to avoid sort of catastrophic forgetting, or we want to avoid potentially overwriting something important in the cortex. And if those memories would be formed kind of online, the danger of erasing something as new memories is formed potentially is high. During sleep, a brain can replay both new memories and old memories together in some potentially weird mixture, and that can help to both map those new memories from hippocampus to cortex, but also to avoid forgetting old memories which have been already represented in some synaptic traces in the cortex. So that's at least one view why it may be happening. So this is a sort of cartoon which illustrates the same idea. So this cute mice, it's sort of in a wake state when it learns something. In the hippocampus, let's say it's moved from the maze and placed cell fire in particular order. So that order repeats, and for some plasticity mechanism, for example, spike timing dependent plasticity, synapses change their strengths between those cells. And that's sort of initial phase of learning. And then during sleep, when there is no sensory input anymore, the same sequences are played back. Some studies suggest they play back at higher rate, but again, still to some extent controversial. And that replay also maps to replay in the cortical cell. So this is a sequence, and this is, so it goes to the cortex. So essentially, cortical cell would replay in particular order and also change synaptic weights between them. So essentially, at the end of this consolidation phase, you sort of map those synaptic traces from hippocampus to the cortex. And this is how they could be stored for as a long-term memory. So this is actually real data illustrating what was just shown from Matt Wilson, which shows this is cortical network, and this is hippocampal network, and this is during awake state when cell fire in particular order, and during sleep the same cell replay in the same order. And it's the cortex and hippocampus. This is recording from hippocampus doing the same. So essentially, that's one of the first study which led to this phenomena, which is called now replay, and which believed to be sort of important element of consolidation. Now, what we're trying to do in our studies, we're trying to use computer models to understand how this consolidation can work, and why particularly some rhythms are important. And because again, lots of data which shows that, for example, if somehow precise coordination between sleep spindles and slow oscillations is disrupted, it will affect negatively consolidation. Or if sharp wave ripples generating hippocampus, which I'm not really going to talk much about, suppressed, it's also impact consolidation. So the question is how all these different structures interact, and why all these rhythms are important. So again, my lab mainly doing computational work. And we use both animal data. This is particularly from Jean-Marc Fillou's study, when rat is learned to learn particular sequence of those places. And so they do recordings in cortex and hippocampus at the same time. This is human studies. This is data from Eric Hallgren and Sid Cash, our sort of main collaborators on human side. And this is intracranial recordings from human patients, epileptic patients. So that's sort of array of electrodes. And also there are some laminar recordings. So all this is kind of data which we are trying to use to constrain our models. Now, to understand, I mentioned earlier that real brain does the sleep stages, and while lots of computational works have been done to understand, for example, how sleep spindles are generated or how slow oscillations are generated, if you really want to try to understand sort of consolidation from learning to sleep and back to awake state, we need somehow to implement those stages. And this is, I'm not going to talk much about this, but this is maybe a very basic element of models, very critical, which we have now, which allows to model transition between different sleep stages and between sleep to wake. Now, again, there are very complex system of nuclei in brainstem and basal forebrain, which release different type of neurotransmitters. And they sort of balance of those is quite different between awake and sleep. There are different models of that. And for example, acetylcholine is high during awake, but drops down during non-REM sleep, increases again during REM. And there are many other changes. So what we did and what was, I think, critical for our studies, memory and learning, is actually to develop a model of sleep stage transitions, which is kind of summarized in this very complex plot. So what I show here is it's a thalamocortical model. So it has a bunch of pyramidal cell, inhibitory interneurons, thalamic relay reticular cell, and we are not modeling explicitly neural circuitry representing those subcortical structures. But since we know what are effect of neuromodulators on intrinsic currents, on synaptic connections, we can actually implement it. And what is shown here is a sequence of transitions from sort of awake state when cortical cell fire randomly. It's very, very, very boring awake state, but it's reasonably characteristic of what is known. Then there is a period of stage 2 when all these things are isolated spindle events. And if you zoom in, this is how spindle looks at LFP, and this is actually this is recording from TC cell, model TC cell. Now, then this is stage 3 when we have up and down states, up state, down states, which again not perfect, but somewhat remind dating back to actually this is a period of REM sleep and then awake state. So this is a model we are using. And why it's important, because when in the following of, in the rest of my talk, I will be talking, for example, training. I will be doing it in awake state. And then the model can be put in sleep state, and we can look what actually those rhythms can be doing. Now, one particular task which sort of inspired to some extent this work is this multi-sequence learning task. This is recordings, again, in human patients done by Sid Cash and Eric Hallgren. So these intracranial recordings. What was shown in this study, the subject was trained to learn certain sequence of finger typing. And what was analyzed is high gamma power across those electrodes. And then some sort of basic classifier was done to look at those patterns during training, how often they happens before, sort of compare how random they are, how often they happens during sleep following this event. So again, this finger typing task, recording during that, high gamma power. So this example, for example, high gamma power could be high here, there, and so on. So it creates some pattern, some sequence. So what was shown that those sequences which appeared during training, during learning the task, actually had a higher than random chance to appear during sleep post-training. It's number one. And number two, those subjects who sort of show more of those replays actually did better in on the test. We showed more replay during sleep after initial training, did better on the task during following awake state when it was tested again. So this is, in a sense, this is a sort of human version of replay, the way I like to put it, because again, it's not a single cell resolution, but it's still a sequence of events which happen during training and repeated during sleep and correlated with performance. So, so this is one reason we use a task, because it's generally believed to be, at least to some extent, hippocampus independent. So this is type of sort of procedural memory, and what I'm going to show at least next few slides would be hippocampus independent. So this is our network, and what we did, we tried to implement that simple task, so in a very simple way. So this is our cortical cell, and we wanted to train it to remember that sequence. So essentially, it was a sequence of inputs to a particular cell, A, B, C, D, E, closely located, and they fire in particular order in response to this input, and that was our training. Now, testing was presenting this just stimulus at position A and see how well this pattern would be completed. So it's kind of pattern completion task. So for example, initially, before training, all synaptic weights are sort of random and not particularly organized, and probability to complete this task, sometimes by chance, if you stimulate at A, it can sort of complete the sequence, but mostly it's not. So that's pre-training state. Now, this is sort of a protocol we use in the model. So it's a wake state. We train that sequence. We can put the model to different stages. It could be just stage 2 sleep or stage 3 sleep or mixture of those. And we can wake it up and test it again. So that would be so this is an example of spindle during stage 2 and slow wave during stage 3. So essentially, it's summarized kind of our protocol. Now, this plot, which I believe maybe Steven did back in a while ago. So we kind of still like it because it shows a lot, kind of very nicely difference between naive and between model which was in sleep state or not. So this is, again, the same protocol, just stage 3 sleep. And essentially, what was done here, this is performance as a result of training. And then one model shown in blue had no sleep, and red model was put in this stage 3, like sleep state during that period. And then performance was tested again when model was waked up in a wake state again. And what we saw that performance of the model, which was during that period of time in sleep state, was improved, significantly improved, while performance of the model, which was staying in a sort of awake state all that time and no training, was slightly reduced or stays the same. I'd like to emphasize the training only done during initial phase. So here, again, was no training during sleep, and during test, we just tested again. There is no additional training. This is some summary of different experiments when we had during that time as stage 2 sleep or stage 3 or mixture of those, this increase in performance. And you can see that performance, it's kind of, in our model, it's baseline performance, around 20. So it's sort of probability of completion of the task. If there is no sleep in the wake state, there is no significant change. And if there is a different sleep stages, there is significant change in performance. And if we can look at synaptic weights in this area, which is an area of interest, and we can see that during sleep, actually, this is during training, obviously synaptic weights increase, but during sleep, they increase as well. So that's why we have performance increase after training, after sleep. Now, the question is why, what happened? So that's kind of what we try to use a model. We try to understand what's going on during sleep, why sleep spindles or slow oscillations may be important to improve performance. And this is an example of area of the network during slow oscillations, which was trained. And you can see that outside that area, this is one of the up states. So this is down state and up state when cell spike, and this is down state again. This is cell index, this is time. And you can see that outside this area cell, generally, the timing of neighboring cell is almost random. So if you pick up a particular cell, another cell, on one cycle to have positive timing, another negative timing, and generally STDP wouldn't lead to any significant changes between those cells. However, here we have what we call replay in the model when a sequence of spikes appears repeatably. And according to STDP rule, which was mentioned earlier this morning talk, if cell fire in particular order, this cell fire earlier and this cell fire later, synaptic connection between them will be improved. So if you look, so essentially what we predicting is that during upstates of slow oscillation, in the area which was trained, cell fire in the order of training. And if you look at the fraction of those sequences which have been replayed, we can see that during awake state, there is not really any replace. And during different sleep stages, there is a substantial amount of replace when, again, cell fire in particular order, and that leads to synaptic changes. Now, importantly, this is whole network. This replay only happens in this area. This is index of the cell across the network, and this is, roughly speaking, amount of replay. So it only happens in the area which was trained. So outside that area, like I said earlier, there is not much replay going on. So the first conclusion I would like to make is that sleep promotes replay of the spike sequences learned during training and awake. So during up states of slow oscillation, replay happens during stage 3. Replay happens during up states of slow oscillation, and it's also happened during spindles. However, during awake states, there is no replay. So next question I wanted to ask: what if you train few sequences, and can they interact somehow? Will they potentially interfere to each other during replay? So we did basically same experiment. Now we pick up two groups of cells, and we train them kind of to learn opposite sequences, A, B, C, D, E, and this was going opposite direction, located, this is real cell indexes located not too far from first one. So it's a complex slide, but just one thing I would like to mention. So if you look at, and what we did also, we wanted to make one memory stronger, another is weaker. So this is now whole network. This is sequence 1. We train A, B, C, D, E, and this is another sequence of train opposite direction. So we train first one longer, which potentially leads to stronger. This is performance right after the training, this middle bar. And you see it's higher for sequence 1, which train longer, and lower for sequence 2, which train less, which is expected. Now, if you look what happens after sleep, when in this case was only stage 3 sleep, only slow oscillations, you can see the first sequence improve a lot. So there are lots of replays of that sequence, and you can see it actually here, this nice sort of organized stripes of cell spiking. However, sequence 2, which was trained in opposite direction and was somewhat inferior to the sequence 1 in a sense how long it was trained, it's actually, there is some quantitative increase, but it's not significant. And if you look at this area, it's not really showing some nice patterns of replay. If you look at actually on how the fraction of replays during those times, what you can see is that for sequence 1, most of the replays happens in correct direction, so from low indexes to high indexes, just like sequence was trained. However, for sequence 2, so this is correct, this is reverse replay. We compare replay in that direction versus replay in opposite direction. So for sequence 1, most of them correct. For sequence 2, actually, most of them are incorrect. So obviously, this replay of that sequence somehow interfere with replay of another memory. And as a result, we have no significant improvement in performance. In fact, if you look at synaptic weights, just ignore those. This is synaptic weights changes. So this is training, and for both sequences, it increases. Red is sequence 1 and purple sequence 2. Then during sleep, sequence 1 continue to increase. For sequence 2 is actually during sleep, it starts to decay at some point. Gray shows what happens if we have sequence 2 alone, so no sequence 1, and in this case, sequence 2 would be increasing. So it kind of suggests that alone, weak sequence 2 will survive. But if we have strong memory trained not so far in a kind of neural estate population from sequence 2, it will interfere and potentially lead to extinction of that memory. So. However, as I mentioned earlier, stage 3 usually happens after stage 2, and there are stages of sleep. And what I showed before was pure stage 3 slow oscillation type of activity. So what happens if we actually add stage 2 sleep before stage 3? So now it's the same exactly protocol, sequences trained to the same amount of time. But now we have period of stage 2 sleep followed by stage 3. And so what we have here, this is sequence 1, this is sequence 2. So there is, again, some increase. In this case, increase in sequence 2 was after initial training was quite low. But what is important, after entire period of sleep, when we train it here, both sequences improve their performance. Well, both memories improve their performance. If you look at the number of replays overall during entire period, now we have more correct replay for sequence 1 in this order than reverse, and the same for sequence 2. It's also replayed in correct order more often than in reversed order. And you can see it here. This is a stronger sequence, kind of. This is the right direction for that. And before you saw that replay of Z in that area interfere with replay of second memory, but now it's not. So this second memory just replay just fine in direction how it was trained. So if you look at the synaptic weights, this is what happened before, but now you can see that during stage 2, there is increase in synaptic strength representing sequence 2, and it's continued to increase during stage 3. So what it suggests, there are two things. First of all, if you have weak memories, they potentially can be extinct. So if there are interfering memory, that interference from stronger memory can negatively impact weak one. So that may happen, which otherwise alone that weaker sequence would survive. However, it shows that if somehow that sequence managed to increase its strength, and it happened during Spindle, during stage 2 enough to sort of exceed certain threshold, then during even stage 3 sleep, both of sequences would be replayed in correct order, and both of them will increase their strength. So what it suggests that having actually stage 2 sleep before stage 3 is somehow useful to try to minimize interference. So why is it? It's still somewhat open question. We try to look at different perspectives, and this is just one view on this. This is a typical spindle, and this is a typical slow wave, and one in the model. And one thing I would like to mention, which kind of obvious, that if you do some clustering on these groups of neurons, spindles are much more independent in a sense. So there are much more kind of local spindles which involve small population of cell, but not really propagating from one group to another. While for slow waves, it's much more global kind of traveling wave type phenomenon. So this number of clusters, for spindles, it's much higher. For slow waves, it's really just 1 or 2 clusters. And spatial correlation drops down much faster for spindles versus slow oscillations. So what it suggests that this local nature of spindles potentially allows to replay multiple memories independently without interference. And once they replay enough to become strong enough, they can survive slow oscillations, which is more efficient way to improve memory because it leads to much faster improvement in replay. But it's also dangerous for weaker memories. Now, this local, does it kind of what I mentioned about local structure of spindles, is it has any grounds in the data? This is human recordings, and it's kind of complex slide. It shows EEG recordings in multiple electrode, and this is MEG data. And why we try to contrast them, because again, not going to find the physics of how EEG and MEG is generated, but presumably EEG pick up more kind of global, more synchronized activity, and MEG looks more in depth of the cortical tissue. But one thing is, you can see that in EEG, this is what is shown here is basically its power of spindle frequency band. So each of this peak would represent spindle. So if you look at the EEG, you see there are very sort of isolated, characteristic big spindle events, and when they happen everywhere. MEG also shows them. But in the deep layers, there are also quite a bit of local events which are not synchronized. For example, it could be some event in one group of electrodes and not so much on another one, and not showing up at all in EEG. So it kind of suggests that spindles indeed, at least in deep layers, can be quite local and less synchronized. Let's keep this slide. So essentially what the second conclusion of my talk is that when memory interference can be introduced, so there are multiple memories which just have been trained and they can interfere to each other one way or another. The weaker memory can only consolidate if it has high enough sort of synaptic strength which represented. And in that case, slow waves are not the best type of activity to actually consolidate that memory because it will favor stronger one. But however, if we have stage 2 and spindles before, it can solve that problem, and it can protect weaker memory and improve performance for both memories. Now, in the remaining very short time of my talk, I would very briefly talk about more recent project which involves stimulation. So this is some of the slides have been discussed already, and basically idea behind it that the different types of sensory cues are known to promote replay and consolidation. It could be olfactory cues, it could be some auditory stimulation. For example, 2 studies I would like to mention. This is from Jan Born group. What it shows that stimulation, auditory stimulation applied in a phase with slow oscillation can actually improve performance on memory task which was trained before. There is an interesting story behind it which I don't have time to discuss, but essentially if there is a kind of closed loop phase locked stimulation, it can help. This is another group of studies which shows that if sort of location of the picture in this field was associated with some sound, and that some of the sounds are replayed during sleep, then those objects which corresponds to these sounds are actually recalled better. For example, in this case, it would be cat. So cat could be recalled better after the sleep. And also, more recent study shows that it actually works better when this during sleep stimulation applied during particular phase of slow oscillation. This is sort of that slow wave I mentioned several times, and they show somewhere around downstate would be an efficient way to stimulate it. So very briefly, just a few slides. So this is same protocol. What we try to do, we try to, in fact, it only shows part of the network. We kind of model, in more recent version, we model also sort of idea of some sensory cortex, which projects to this network. And what we did here, we, again, train the sequence the same way, and there is a period of sleep, we test performance. But now we also apply stimulation. In this case, it was directly applied to this network, but in more recent versions, we stimulate actually the sensory cortex, which projects to randomly and sparsely to this sort of motor cortex. Essentially, what is shown here is if we train network very weakly, we can get situation when improvement during slow wave sleep alone could be very small. I mean, it's smaller than what I showed before just because I train network less in this case. So it's not trained enough. It shows some improvement, but it's almost non-significant. However, if we add stimulation, it's actually improve significantly performance of completion of that sequence. Now, interestingly, that improvement is not related to high number of slow waves. We actually did control for that. And yes, stimulation can increase slightly frequency of slow waves, but if we compensate for that, reducing duration of sleep, we still have improvement. This is basically this case corresponds to this one corresponds to this one, and this is a case when we reduce duration of sleep in case of stimulation, just to show that something else happening which helps to improve performance without stimulation. The phase of stimulation is matters, just like it was shown in data. So this is case when we use closed loop stimulation. So again, we stimulated our network. We stimulated actually sensory network which projects to this one. And we could pick up different phase of stimulation. This example when we stimulate just at the end of down state before transition to up state. And we can see this is the best case how we can entrain the network. This is initiation sites for up state. You can see this peak shows us if we stimulate it this way, we have very high probability to entrain the network to initiate new up states. And performance increase also highest. If you stimulate, for example, during up state, when network already in up state, it actually doesn't affect, as we can expect, initiation sites, and performance change is low. So it suggests that the phase, as was already shown by experimental data, is matters, but it also suggests that it's because stimulation at right phase can initiate those slow waves, and as a result can promote replay of those sequences we have been trying to learn in the network. My last slide, what happens if you have two sequences, and this is very recent data just from earlier this week's analysis. What has shown that if you look, for example, at this case, we have two sequences which we train, one is stronger than another, and we try to apply Q to affect one versus another. So it just shows that we can get different combinations. So for example, if you have stronger sequence and Q it during sleep, you improve it significantly, and you can almost suppress completely the second memory. However, if you Q the weaker one, you improve it. Now this is this weaker sequence which train less. You improve it, but it almost doesn't affect the stronger sequence. So it suggests that if sequence is strong enough, if you, again, there is a replay of something else, Q it or not, that sequence will improve during sleep, and that memory can improve. But if memory is weak, it's potentially more vulnerable to interference, and then, for example, trying to Q stronger sequence can lead to almost complete extinction of the weak one. So, so it suggests that local stimulation presented in a specific phase of the slow oscillation maximize memory performance, and depending on protocol, it can either increase or disrupt consolidation. Now, there's a long kind of conclusion, but I guess the only one point I would like to bring from here is that, interestingly, natural sort of cycle of sleep, with sleep stages going away from sleep spindles to slow waves and repeating several times that way seems to be important for protecting memories from interference. So these two people who currently in my lab did most of the work. I already mentioned Steven did one, started that project, so it's kind of moved from off the ground. We collaborate with many experimental labs, and this is our support. Thank you.
I have two modeling questions. What do you do to your model to make it switch between the stages? You're doing some change in...
Essentially, we model effects of neuromodulators. So for example, we know that acetylcholine reduces from wake to stage 2 to stage 3. And we know that it changed potassium leak current, it changed AMPA conductance between cortical cell. So we implement those changes. So here we model it specifically acetylcholine, norepinephrine, and change in GABA. So those three was kind of minimal model which was sufficient to explain transitions. I'm not saying that it's the whole picture, but that's how we did it here.
Right. And then when you do the stimulation, I mean, it seems the Q is, not a direct, pulse of current to a neuron and so on. But is that how you're modeling it?
So in the early versions, this is how we modeled it. What we do now, we have another kind of network which represents the sensory cortex, which initially sparsely connected to the network I show you. And during training, we stimulate some site of that network as well. Now, because there is a timing between that sensory network and sort of motor network, connection increase. And during sleep, when we stimulate the same site in the sensory cortex, it helps trigger slow wave or spindle or something. But in general, I would say that's kind of not, it's difficult to model it at the level of complexity. It should be modeled, but we kind of move away from single point stimulation, but that's, yeah.
So I'm particularly interested in how the long-term consolidated memories that are already in the neocortex might also be reactivated and contribute to efficiently integrating new information. And I wondered if you had any thoughts about that, or whether you've looked at those kinds of
The main one, it's something we are very interested to look at. It's a very interesting question. So we try to look at certain things. For example, if you train one sequence in the certain order, and if it's kind of embedded to the cortical network, and then we try to train a new one, which would be close enough and somehow not orthogonal to the first one, then in general, that second one would be replayed better, simply because replay of the first memory, it happens spontaneously. Even without any stimulation, if you have embedded memory to the cortical network, some connection are changed, that sequence will like to repeat during up states. So if there is another recently learned sequence which kind of close enough and in right direction, then replay of the first one will help replace the second one. If it's kind of in opposite direction, sounds like its effect is opposite. So in a sense, I would like to interpret this as saying that if something has been learned in the cortical network, replay of those memories can help to consolidate information which is similar or close or somehow related to the first one. And if it's not, then effect may not be that much. But that's a bit of stretch of what we have now. So we are trying to explore it, and we have very early data, but yeah, that's how we cannot see it. Yeah. Yes.
So, beautiful, really interesting. So, when you say replay a sequence, like in my naive computational approach, like we do this thing where we generate new inputs, which is like, again, we generate external thing in the world, not stuff really in the brain. Something that people like will like to discuss it would be like, would we be able not to generate the whole input and then propagate the input through our, like through the whole cortex, but directly generate activity in particular point of the cortex. So maybe particularly it's related to James' idea. So like, where should, do you think like, like if there is a cortical process that takes a lot of the cortex, but maybe only some parts of the cortex need to be modified.
Exactly.
Some part or needs to reduce.
Right. So that's in a sense what potentially distinct between AI-type networks and biological networks, because presumably, if you go back to, let's say, whole picture, including hippocampus, right, and there is something new is learned and represented in hippocampal structure, then essentially only part of the cortical network which represents same category or same type of the same class of the object need to be replayed. You don't need to touch anything else. That's why you can more easily to avoid catastrophic forgetting in biological networks, because in principle, connection from hippocampus to the PFC through, let's say, sharp wave ripples shooting from hippocampus and initiating upstates in the cortex, as we're trying to explore in some other study, will trigger replay exactly in the area of the cortex which needs to be replayed to consolidate new memories of a particular category. So if you want to learn another type of car, you kind of replay only maybe categories of car somewhere in the cortex. You're not touching planes or something. If you want to create new categories, then it's more difficult task. But again, not whole cortex needs to be replayed. So again, that's not this obviously a simplified picture in the model, one-dimensional network, and it seems like it's propagating through. But even here, as you can see, replay only localized to particular area of the network. In fact, that paper we have reviewers specifically asked the question. It looks like it propagates through entire network. Everything is going to be replayed. But even here, if you look at the cell outside brain area, they are not replayed. They are not changed as connections. So whatever was represented there will stay there. But again, the whole picture probably involving this, this magnetic categories, this pyramid of categories and so on. It's more complex, but yes, so that's what we believe. So this hippocampal down to cortex kind of structure allows to replay only what needs to be replayed to kind of memorize new information, not more than that. Okay.